Hello, everyone. My name is Abraham Vargas. I'm a Project WiCCED mentee at Delaware State University, and I would like to present my research on express sequence tags linking salt tolerance in Spirabolus alterniflorus to Triticum aestivum. So the main objectives of this research was to see how we would develop more improvements of making staple crops such as bright wheat more sustainable resources in high salinity conditions due to rising saltwater levels moving more inland. By looking at the familiar species in its family line, we believe that any similar mechanisms or genes that help combat salt tolerance would help us understand a little bit more of how a solution may be possible. The different methods used in this research was the National Center of Bioinformatic Information, also known as NCBI. Using the National Center for Bioinformatic Information, we collected relevant data from journal articles and backtracked genes mentioned in these articles to seek out any correlation between Spirabolus alterniflorus and the Triticum aestivum genome. So for this part, we would create a theoretical experimental design. The methods used for this theoretical experiment would be primer design to first seek out if the INO1 gene, which is the specific gene that we want to look at, is in fact inside the Triticum aestivum genome. For the next part of this experiment, we would use a transgenic process or method called biolistics, also known as the gene gun method. This binds DNA to small particles of gold or tungsten inside a chamber in which it is shot into the plant cell or tissue with high pressure. The particles will assimilate through the cell wall and the cell membrane into the genome of the plant, and the DNA will separate from the coated metal. So the specific DNA strand that we would want to insert in this experiment would be the INO1 gene. So in the results, we'll explain a little bit more about what INO1 does. So EST, or express sequence tag 2557, correlates the INO1 gene in Spirabolus alterniflorus to a gene called MIPS, which is myoinositol 1-phosphate synthase. This is a rate-limiting enzyme that catalyzes the reaction from glucose 6-phosphate to MIP, the first step in myoinositol biosynthesis, in which an upregulation in the pathway would lead to an increased inositol production. So myoinositol has always been involved in salt tolerance of many plants as a facilitator of uptaking and long-distance transport of sodium. Factors such as proline in the carotid content, root length, and as well as qualitative factors like dead and browning of leaves were measured for signs of myoinositol overexpression. The figure on the left shows how exactly the gene gun method works, and the figure on the right shows like a glimpse of the theoretical experiment that we would design with 5 different salinities or however many different salinities, and seeing how these transgenic plants grow or develop in these salt conditions. The rest of the results just go into the comparison of homologous genes of Spirabolus alterniflorus to Triticum aestivum and the similar mechanisms that they have. So concluding, by comparing these two plant genomes from the same family, we hypothesized that the INO1 gene would develop a similar stress defensive mechanism, as shown in the results of EST2557 in Triticum aestivum, for the reason that there are a number of homologous genes in both species that have this same, or if not similar, mechanisms for stress shown in the other sections of those results. We would set up an experiment using transgenesis to then use those results to confirm or deny the hypothesis in a future study. And I would like to give a special thanks to all the Project WiCCED mentors and the cyber director, Dr. Cal, for all the help and guidance throughout this experience. Thank you.